Let's look at the nervous system 2, neurophysiology. So we will talk about some membrane proteins, including the pump, sodium-potassium pump, and calcium pump. We also will talk about the channel, the leak channel, sodium channel, potassium leak channel. And these pumps, sodium-potassium pump, that's the one maintained in the resting state. Sodium-potassium pump will keep pumping sodium out, potassium in. So it keeps the environment of high sodium outside, low potassium inside. Calcium pump, that's the one to keep pumping calcium out of the cell. So inside the cell, calcium concentration is pretty low. And the leak channel is to make sure that don't overdo it. So still you have a normal range of sodium, high sodium outside and high potassium inside. So this is before the action potential happens. And these chemical gated channels, chemically gated sodium, we're mainly talking about this, and potassium and chloride. This channel happens in the dendrite part. When other neurons come to talk to this neuron, they will release neurotransmitter, and neurotransmitter will open those chemically gated ion channel. So mainly the sodium. So when you have more neurotransmitter release, more sodium channel open, and when the sodium channel open, sodium flow in and can make the inside more positive. That's how they start to create we call the graded potential. And start from the axon here lag, it will generate action potential. So this graded potential start from the dendrite, go to the cell, and eventually the summation of the voltage change, if they're bigger than the threshold in the axon here lag, that's the place they start to have a lot of voltage gated sodium and voltage gated potassium channel. So it will open the voltage gated sodium channel and potassium channel. So what happens is action potential is generated. Start from the axon here lag. It will go through the whole axon, reach the terminal part. When you reach the terminal part, you have the voltage gated calcium channel waiting there. And this calcium channel will make the calcium flow in, and it will trigger neurotransmitter release. So that's the place it will release neurotransmitter and will activate the other cells dendrite, and the electrical signal keep going. So that's the neurons, and this their distributions. Those voltage gated ion channels start from the axon here lag to the terminal. In the terminal part, they have the voltage gated calcium channel. So this is the voltage gated sodium channel. It depends, it sends the voltage change, and when the voltage change, it will open. Once it opens, sodium gonna flow in because sodium is high outside, low inside. When the sodium flow in, make the inside become more positive. And that's how they create action potential. And the voltage gated potassium channel, once it's open, potassium gonna flow from high that's inside and out. So once it flow out, it will cause the repolarization. You found the same concept as you learned in the muscle physiology, also apply here in the neuron part. And when the neuron is in the resting state, inside is about -70 millivolt. And they have the chemically gated ion channels surround the dendrite and also the cell body. In the axon and axon terminal, you have the voltage gated channel. So voltage gated sodium, potassium in the whole axon. And in the axon terminal part, you have the voltage gated calcium channel. When the cell is in resting state, the voltage we call the resting membrane potential is about -70 millivolt. As due to the sodium potassium pump, it keeps pumping sodium out, potassium in. And once you have too much, you have the leak channel, you have the potassium leak channel, sodium leak channel to maintain the concentration. So your resting membrane potential is very stable, about -70 millivolt. And these are some terminologies like the voltage, that's the separation of electrical charge, and when they start to flow, we call the current. And resistance is how easily or how difficult they can move through. So apparently you can open close certain ion channel, you can increase or decrease the resistance. And when the action potential go to the terminal part, so this is very similar to the motor end plate. So this is in the muscle part, we call this motor end plate, and this is the motor neuron, and the same concept applied to the neuron part. So we have one neuron connect with another neuron. In the presynaptic neuron, they need the calcium to trigger the neurotransmitter release. So in the motor neuron, this is acetylcholine, but this time we just have 11 different kind of neurotransmitter, depends on what neuron it is. So still calcium flow in, you open the voltage gated calcium channel, calcium flow in, and calcium will trigger the vesicle fusion. Once they fuse together, neurotransmitter release. And this we call the postsynaptic neuron, and they have the neurotransmitter receptor within there. These are the ligand-gated ion channel, most of them sodium channel. When the neurotransmitter bind with the ligand-gated sodium channel open, then sodium flow in, and the sodium flow in will make the inside become more positive. So in the muscle part, you learn this is called the end plate potential. But in the neuron part, we call this EPSP, excitatory postsynaptic potential. But exactly the same concept. So what happened in the connecting part, because of the synaptic gap, the small gap, the electrical signal need to be transferred into the neurotransmitter release. And you have excitatory synapse. Most of them are excitatory synapse. So these synapse, they have a lot of ligand-gated sodium channel. When the neurotransmitter is released, it will bind with the sodium channel open, sodium flow in, inside become more positive, and these are the excitatory. You also have inhibitory synapse. So this time, these receptors are actually chloride channel. So chloride is also high outside. And once you release the neurotransmitter, example one is called GABA, G-A-B-A, GABA. And when you release GABA, you will bind with the chloride channel. And when the chloride channel open, chloride flow in. And chloride will make the inside less positive, we call the IPSP, inhibitory postsynaptic potential. And inside become more negative, is less likely to generate an action potential. So you have excitatory synapse, inhibitory synapse. And these graded potential, they are good for local signal, very similar to the end plate potential in the muscle. It's good for the local, but if you want them to travel for long distance, it's like the wave, it will gradually decrease. So this won't be able to travel for long distance. In order to travel for long distance, you need to generate action potential. So that's graded potential. You start from this point, and through the distance, it gradually decays. So it's good for local, but not very good for traveling for long distance. And the purpose of this graded potential is to make the signal big enough. So if you open the ligand-gated sodium channel, it depolarizes, like sodium flow in, and make the inside more positive. We call it excitatory postsynaptic potential, EPSP. If you open the chloride channel, what happens is chloride flow in, make the inside more negative. We call it IPSP, inhibitory postsynaptic potential. And we call this hyperpolarization. And EPSP, make the inside become more positive. IPSP, is the inside more negative. And eventually, it's all the summation of those EPSP, IPSP, and equals the depolarization. So most of them create the EPSP. Most neurotransmitter create EPSP. Some of them create the IPSP. And this one create the IPSP. You open the chloride channel, that's the main one. You can also open the potassium channel because potassium is going to flow out. So it will make the inside more negative as well. So that's the IPSP. And on average, every neuron receive hundreds of synapses, if it's not thousands. So every neuron in every seconds, they actually receive a lot of neurons yelling at this neuron, ask them fire, don't fire, fire, don't fire. And eventually it's the summation of all the EPSP and IPSP. Depends on does it reach the threshold. So neurons use a lot of energy. Your brain uses a lot of energy. And the summation of all the EPSP, IPSP, if it go over the threshold, what we care is in this area, we call the axon hillock, because that's the area you have a lot of voltage-gated sodium and potassium channel. And these dendrites, they can have the spatial summation because they have so many different dendrites. And even though each EPSP is small, but they can sum up together, they can reach the threshold. So if you go over the threshold of the voltage-gated sodium channel, it can create action potential. We call this spatial summation. And it can also have the temporal summation. The same synapse, it can trigger once, even though the signal is small, but it can trigger more than once, so they can sum up together. We call this temporal summation. And all the summation together, if they are below the threshold, we call them sub-threshold stimulus, and that's nothing happens, so no action potential. If you go over the threshold stimulus, it's enough to generate action potential, and it will generate the action potential. So the action potential is all or none. Either you have 100% action potential or you have nothing. You start from the axon here lag, it will go through the whole axon, very similar to the action potential goes through the whole T-tubule in the muscle. And the whole axon covered by the voltage-gated sodium and potassium channel. So it will cause the depolarization, repolarization, it will go through the whole axon and reach the terminal. So action potential can travel for long distance. And these slides show you how to generate action potential. First, you need to have the EPSP go over the threshold. Then the voltage-gated sodium channel open, cause a big depolarization. Then the voltage-gated potassium channel open, cause a repolarization, and it go back to normal. That's the action potential. It takes about 2 to 3 milliseconds to finish. And these slides tell you the same thing. You have the voltage-gated sodium channel open, and sodium flow in. It causes a big depolarization. That's this part. And sodium channel close, potassium channel open. Potassium channel open, potassium flow out. So it generates one complete action potential. And now this action potential can trigger the opening of the voltage-gated channels close to it. So now the action potential can start to walk through the whole axon. And the action potential takes a very short time to generate. And the refractory period, that's the time these ion channels are busy to work on this action potential. So they could not generate another action potential. So we say the refractory period is very short in the neuron. And we have the absolute refractory period, so there's absolutely no way to generate action potential. And we have the relative refractory period. That's the time if you provide a bigger than normal stimulus, you can still open certain ion channel because you have some ion channels that are ready. That's the relative refractory period. So overall, the refractory period of neuron is very short. So it takes them a very short time to generate another action potential. In AP2, when your instructor talk about the heart muscle refractory period is very long. So it's a protective mechanism. Your heart muscle won't be able to have muscle cramp because they have a super long refractory period. So again, that's the action potential. Start from resting and the depolarization. This tell you what ion channel open, voltage-gated sodium channel open, and voltage-gated potassium channel open cause the repolarization, and hyperpolarize a little bit, go back to normal. Okay, let's take a break.